Hello, everyone, and welcome to The Explainer. I am absolutely thrilled to have you with us today because we are about to go on a seriously fascinating journey. We're going to dive deep into the microscopic world right beneath our feet, a world that honestly holds the key to one of the most pressing health emergencies of our entire lifetime. We're going to break down exactly how human history, cutting-edge technology, and literally the dirt in your backyard are all perfectly interconnected. So, think about this for a second. What if the cure to our greatest medical threat is actually buried in the dirt? I know, it sounds kind of like science fiction, right? We've got these massive, multi-billion-dollar pharmaceutical labs all over the world. And yet, the ultimate solution to highly advanced, drug-resistant superbugs might just be waiting for us in a literal handful of soil. To really understand why this is so absolutely crucial right now, we gotta look at the numbers. 4.95 million. Take a moment to let that sink in. This isn't just a random statistic. That number represents the staggering global death toll associated with antimicrobial resistance in the year 2019 alone. We are talking about nearly 5 million lives lost just because the bacteria infecting them have evolved. They've figured out how to survive the very drugs that were designed to kill them. This is the heavy, heavy reality of the data we're dealing with today. Okay, let's dive right into this. Part 1, the resistance crisis. The massive gap we're dealing with brilliantly illustrates the terrifying deficit we are currently facing in modern medicine. In a single year, the World Health Organization tracked 23 million bacteriologically confirmed cases of antimicrobial resistance across 104 countries. 23 million! But in the face of that massive tidal wave of infections, we had a tiny, tiny trickle of just 13 new antibacterial drugs approved between 2017 and 2023. You know, it's literally like bringing a knife to a gunfight. And honestly, it gets even more concerning when we look a little closer at those 13 drugs. Out of those 13 newly approved drugs, a mere just two, actually met the WHO's criteria for genuine innovation. The other 11, they did not. Now, genuine innovation means the drug has a completely novel mode of action, or a new target, or it lacks cross-resistance with older drugs. Without those features, bacteria easily adapt. They figure it out. We desperately need new weapons. So, where exactly do we find them? That brings us to part 2, the Golden Age. Let's rewind a bit and look at our historical relationship with soil. Back in the mid-20th century, we were right in the middle of the golden age of antibiotic discovery. Scientists were systematically screening soil microbes and, frankly, hitting the jackpot. They were discovering actual miracle drugs like penicillin and tetracyclines. See, soil microbes naturally produce these compounds to fight off other bacteria in their hyper-competitive dirt environments. It's a microscopic war zone down there. But by the late 1960s, we hit a wall. We experienced this massive discovery plateau caused by what's called the re-isolation problem. Basically, scientists just kept finding the exact same compounds over and over again. Frustrated by this, the pharmaceutical industry shifted gears in the 2000s, moving completely away from dirt and toward purely synthetic drug development in the lab. Which leads us to part 3, the discovery void. So, the absolutely crucial point here is something called the culturability crisis. Turns out, we didn't hit a plateau in the 1960s because we actually ran out of useful soil bacteria. Not at all. We hit a plateau because 99% of environmental microorganisms simply refuse to grow in a standard laboratory petri dish. Those standard agar plates completely lack the variable nutrients and complex ecological signaling molecules that these wild microbes actually need to survive. Think of it like trying to grow a super rare, delicate rainforest orchid in a sterile concrete room. It's just not going to work. We were totally blind to 99% of the biological wealth sitting right there in the soil. Now, to bypass this culturability crisis, scientists tried to invent completely synthetic drugs from scratch. But, well, that largely failed. Why? Because while these synthetic compounds worked beautifully in vitro, meaning in a totally controlled test tube, they lacked whole cell activity against real complex bacteria in the real world. They completely failed to penetrate the incredibly tough outer membranes of gram-negative bacteria. And even if they somehow did get inside, the bacteria just used their internal efflux systems to literally pump the drug right back out. Basically, we learned the hard way that nature is just a far, far better chemist than we are. Enter part 4, unlocking microbial dark matter. Okay, so microbial dark matter is pretty much exactly what it sounds like. It's that vast, unculturable 99% of microbes in the soil. whose incredible genetic and biosynthetic potential has been completely invisible to us up until now. Think of it as a virtually infinite, completely untouched underground pharmacy. We always knew the pharmacy was there, but the front doors were locked tight. Well, today, we finally have the keys. Now, what's really interesting here is this specific piece of tech called the iChip. It's literally one of those master keys I just mentioned. Instead of forcing bacteria to grow in an artificial lab, the iChip brilliantly tricks the bacteria into thinking they never even left home. Here's how it works. First, scientists dilute a soil sample so just one single bacterial cell goes into a tiny chamber on the chip. Second, they seal the device with semi-permeable membranes. And third, and this is the true genius part, they literally bury the device right back in the soil. The natural nutrients and growth factors from the real dirt diffuse right through the membrane. The bacteria grow perfectly in situ, right where they belong, allowing us to finally cultivate and study that previously unculturable 99%. And let me tell you, the results of this new tech are just awe-inspiring. We're already seeing actual recent discoveries that prove tapping into this microbial dark matter really works. For example, look at Teixobactin, which was discovered using the iChip. It inhibits bacterial cell wall synthesis by binding to these highly conserved lipid precursors. And remarkably, researchers found literally no detectable resistance in lab tests against nasty pathogens like staph and tuberculosis. No resistance. Then there's Lariacidin, which binds to a unique site on the bacterial ribosome to stop protein synthesis. It boasts broad-spectrum activity with super low spontaneous resistance. These are exactly the kind of novel, game-changing mechanisms we so desperately need right now. But hey, the iChip isn't the only tool we have in our modern discovery toolkit. We're now using metagenomic sequencing and environmental DNA to analyze soil without needing to grow the bacteria at all. We're using genome mining to identify hidden biosynthetic gene clusters. And we're even unleashing artificial intelligence to rapidly screen millions of virtual molecules based on all this genomic data. It takes this incredible, synergistic combination of returning to the raw nature of soil and applying the world's absolute most cutting-edge AI in genomics to track down these life-saving natural pathways. Which brings us to our final section, Part 5, the double-edged sword. Now, we do have to talk about a very real critical warning here. Human practices are actively pumping resistance genes right back into the very soil we're so desperately trying to study. Through our massive use of agricultural manure, sewage sludge, and reclaimed wastewater, we're introducing antibiotic residues and resistant bacteria directly into the earth. And guess what? The soil microbes are adapting to this pollution. They're exchanging and hoarding antimicrobial resistance genes, or ARGs. So the soil isn't just an untouched pharmacy anymore. We are actually turning it into a training ground for superbugs. All of this research points to one incredibly powerful conclusion. Quote, soil must be viewed through a planetary health and One Health perspective. It is a reservoir of antimicrobial innovation, but also a pathway through which resistance determinants can move among soil, water, food, animals, and humans. End quote. I mean, that perfectly captures the whole full circle arc of our explainer today. We simply cannot separate human health from the health of our planet's soil. They are completely intertwined. They are one and the same. So, what does this all mean for us? The question we really have to ask ourselves as we wrap up is this. Can we protect the very ecosystem that holds the secret to saving us from the antibiotic resistance crisis? We finally have the mind-blowing technology to unlock the microbial dark matter beneath our feet, but honestly, it won't matter at all if we don't act as good stewards of our environment first. I really hope this explainer has sparked your curiosity and given you a whole new appreciation for the simple dirt beneath your shoes. Thank you so much for joining me today, and I highly, highly encourage you to keep exploring this amazing topic.